Jai everyone, I am Pankaj Kumar, and this lecture is regarding the DNA replication, part third, lecture third. And in this, we will read about the telomere replication and as well as replication related disorder. So this video will be more exciting than the other videos because in this video we will see the applications based disorder, like what are the actually visible if the DNA replication is not occurred properly, and how we can relate the DNA replication with the life example. Okay, so this is a very important video, and most of the students are saying like why we are reading this biology and again. This is a education system made us to like a ratification, so we have to repeat again, study again things, and ultimately some after reading we will become a professor or some lecturer. Then again we will read teach these things. So what we are actually gaining from this study. So that's why this video is very important. In this, you will understand what is the actual gain of this studies, not only the theoretical knowledge, but actually the life examples by which we can understand why we are reading this topic. Okay, so let's begin with this DNA telomere replication. My dear students and my friends or viewers, so this is the normal DNA strand. This is the leading strand. This is the leading strand, and this is the lagging strand. Now this leading strand is something which is which doesn't have very much trouble during the replication because it need a one primer and it will end till the end of the chromosome, till the end of the DNA. So we have a chromosome such as like this, and the end of this is called as a telomere, where you will find a DNA strand where it is going to be end. Okay, suppose this is the end of this. So this end of the telomere DNA doesn't have any problem in the leading strand. Okay, so where is the problem arise? If you see the lagging strand. In lagging strand, the primers, usually the primer will attach at a certain distance. It will not, every primer will not attach very near to each other, but it will need a certain distance at which the new primer can be added. So this is around 200 nucleotide base pair. Okay, it is around 200 nucleotide base pair. So if a primer is added at this position, so after this it will added around the 200 nucleotide only. It's not going to add in between this. So for the different organism it may be vary, but the actual data is 200 nucleotide for the telomere replication. Now this is the primer which you added, you extended till here. Primer you added, you extended till here. Then you primer you added, you extended till here. But if it is less than 200 nucleotide, if this distance is less than 200 nucleotide, this one, then you cannot add a primer here. You cannot add a new primer here. So you have to add new primer somewhere at this position, suppose. Somewhere at this position. Okay. So this is why this portion of the DNA will be eliminated and it's not going to replicate, and finally you lose this portion and the DNA will be shortened. Suppose again if this DNA is sorting, this is again leading and this is a lagging strand. You added a primer, you replicated, you replicated. Now this region is also again shorter than the actual one. Okay. Now the new DNA strand, whatever you will get up to this length only. You will get up to this length only. Are you getting my point? So up to this length, the new DNA, not for in leading it's not a problem, in lagging only. So up to this length will be reduced. So ultimately what you will find, the DNA will be shortened. Okay, the DNA will be shortened from the actual length of the DNA. So this shortening of a DNA replication, this shortening of the DNA need some mechanism to maintain the length of the DNA. So what is that? We will talk about it, but this is the one way by which the DNA replication will be shortened. Another way is what? Another is a primer. What if this length is up to 200 nucleotide? Suppose this length is up to 200 nucleotide and you added a primer here. Now you people are happy? Very happy. Okay, fine. So whenever you need to remove this primer, the DNA polymerase 1 will come and it will remove in case of the bacteria. But it required this position. It required this polymerase, this DNA polymerase need a strand where it will stand. Okay, it cannot It cannot stand in between the air. It cannot stand in between the cytoplasm somewhere or nucleoplasm. It needs a stand at which it will stand. Are you getting my point? Are you getting my point? Okay. So यहाँ पे तो स्ट्रैंड खत्म हो गया. तो ये जो पॉपेट है, बाबू है छोटा सा, ये छोटा सा बाबू जो डॉल है, ये डॉल तो यहाँ पे नहीं बैठ पाएगा. यहाँ बैठेगा? नहीं बैठेगा. अब यहाँ नहीं बैठेगा तो उसको कैसे इकट्ठा उसको बनाएगा? नहीं बना पाएगा इसको. जब इसको नहीं बना पाएगा, यहाँ बैठ नहीं पाएगा, तो क्या हो जाएगा? गिर जाएगा. और ये गिर जाएगा तो फिर क्या करेगा? चिल्लाएगा, ऐसे करके चिल्लाएगा, और यहाँ पे रेप्लिकेशन नहीं होता है. आगे बात समझ? आगे बात समझ? तो यहाँ पे ये बढ़ा, ये बढ़ा, ये बढ़ा, लेकिन यहाँ पे तो ये छूट जाएगा. यहाँ पे ये क्या हो जाएगा? छूट जाएगा. सो अगेन स्लोली ये क्या होगा? छोटा हो जाएगा डीएनए. यहाँ पे डीएनए क्या हो जाएगा? क्योंकि वो छोटा छोटा कट जाएगा, ये कट जाएगा, ये कट जाएगा. क्यों कट रहा है? क्योंकि डीएनए पॉलीमरेज 1 बाइंड नहीं कर पाएगा. उतनी उसके पास जगह ही नहीं है बाइंड करने की. मेरी बात समझ में आ रही है? यहाँ से कोई दिक्कत है? इजी है ना? चलो अब हम लोग बात करते हैं कि इसका सॉल्यूशन क्या है. तो इसका जो सॉल्यूशन है, नेचर में भगवान ने इसको क्या दिया है? भगवान बोलो, ईश्वर बोलो, जो भी बोलना बोलो, अल्लाह बोलो, जिसने भी दिया, एक सॉल्यूशन दिया. उसका सॉल्यूशन क्या है कि डीएनए पॉलीमरेज यहाँ पे नहीं बाइंड कर रहा, यहाँ पे, और इस दोनों केसेस में डीएनए छोटा हो जाता है. तो हमें क्या चाहिए? एक बड़ा सा हेवी दाना चाहिए. दाना चाहिए या देवता चाहिए, जो भी हो, हमें एक ऐसा बड़ा सा प्रोटीन चाहिए, एक बड़ा सा दाना चाहिए, जो क्या कर सकता है इसको, एक देवता, एक बड़ा सा दूध चाहिए जो इसको आगे बढ़ाए, है ना? जो यहाँ पे छोटा हो रहा था इसको आगे बढ़ाए. उसको हम लोग कहते हैं टीलोमरेज. क्या बोलते हैं? सो टीलोमरेज एक ऐसा एंजाइम है जिसमें क्या है, इसके अंदर आरएनए भी है और प्लस प्रोटीन भी है. तो इसके अंदर आरएनए जो होता है बहुत लगभग 450 न्यूक्लियोटाइड का आरएनए होता है. ठीक है. तो ये छोटा तो नहीं है नंबर, 450 छोटा नंबर है कि बड़ा नंबर है? काफी ठीक ठाक नंबर है ना? ये जो 450 न्यूक्लियोटाइड का टीलोमरेज है, इसके अंदर आरएनए भी है, और अलग अलग ऑर्गेनिज्म में क्या होता है, ये टीलोमरेज का जो आरएनए का सीक्वेंस होता है, स्पेसिफिक सीक्वेंस होता है और अलग अलग होता है. मैं इस वीडियो में ही बताऊंगा आपको कि वो कौन कौन सा होता है. आगे बात समझ? तो ह्यूमन में क्या है, टीटीएजी, ये पेरेंट स्टैंड है. ये हमारा क्या है? पेरेंट स्टैंड है ना? तो हमारा जो पेरेंट स्टैंड है, जो ब्लैक कलर से बनाया, उसमें टीटीए ट्रिपल जी जो है, ये टीलोमर सीक्वेंस है. ये क्या है? इसको टीलोमर सीक्वेंस कहेंगे. क्या कहेंगे इसको? चलो इसको कहेंगे टीलोमर सीक्वेंस. अभी जो टीलोमर सीक्वेंस है, यहाँ पे ये टीलोमरेज में जो आरएनए है, उससे इसकी कुछ मैचिंग है. इसके साथ क्या है? थोड़ा बहुत मैचिंग है. ये कॉम्प्लीमेंट्री है इस दोनों का. तो ये कॉम्प्लीमेंट्री टीटी की जगह ए बाइंड कर रहा है, ए के साथ यू बाइंड कर रहा है, ट्रिपल जी के साथ ट्रिपल सी बाइंड कर रहा है. इसके अलावा भी आरएनए है इसके में. अब यहाँ पे जब नया आरएनए है तो ये क्या होगा, जो पेरेंट स्ट्रैंड है उसके साथ नया न्यूक्लियोटाइड जोड़ देगा. यहाँ पे टीटी आ जाएगा, यहाँ पे ए आ जाएगा, यहाँ पे जी आ जाएगा. इसी तरह से फिर क्या होगा, आप पाओगे कि जो पेरेंट स्ट्रैंड है वो धीरे धीरे क्या हो जाएगा, बड़ा होता चला जाएगा. पेरेंट स्ट्रैंड क्या हो जाएगा, धीरे धीरे बड़ा होता चला जाएगा. अब यहाँ पे आपने क्या किया, यहाँ पे हमारा नया स्ट्रैंड आ गया था TTAG. अब ये जो हमारा जो स्ट्रैंड है, ये थोड़ा सा और घिसक जाएगा. क्या हो जाएगा? और घिसक जाएगा. CCCAA. ये हमारे पॉलीमरेज का ब्लू कलर से लिखना है. तो ये आ जाएगा CCCAA TC. यहाँ पे U आएगा, UCCC. फिर वापस यहाँ पे और बढ़ गया. ये जो हमारा ये है टीलोमरेज जो है, ये क्या हो गया, और बढ़ गया, है ना? इसको बाल बना दे थोड़ा सा, ये बाल बना दे, है ना? चलो, तो ये क्या हो गया, थोड़ा सा और बढ़ गया. तो इस तरह से बढ़ते 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 क्या हो जाएगा, ये इतना बढ़ जाएगा कि ये जो स्ट्रैंड था, अब ये रुक के यहाँ पे 200 न्यूक्लियोटाइड तक पहुंच जाएगा. तो अब यहाँ पे ये थोड़ा सा बढ़ गया. यहाँ पे एक और प्राइमर हम लोग जोड़ सकते हैं. अब जब ये प्राइमर जोड़ सकते हैं तो वापस हम लोग इसको बढ़ा सकते हैं. तो अब बोलोगे सर ये भी छूट जाएगा, तो इसको क्या करेंगे? तो इसकी हमें बहुत ज्यादा जरूरत ही नहीं है, क्योंकि हमें जहाँ हमारा जो एक्चुअल डीएनए था वो यहाँ तक ही था, हमारा एक्चुअल डीएनए यहाँ तक ही था, इसको बढ़ा के किया गया है, तो ये नहीं भी करेगा तो हमारा जो एक्चुअल पेरेंट डीएनए है वो रेप्लिकेट हो जाता है। आ रही बात समझ, कोई दिक्कत? तो ये टेलोमेरेज रेप्लिकेशन आपको समझ में आ गई होगी। जो इंपॉर्टेंट चीज है वो एक बार हम आपको बता देते हैं। ये जो सीक्वेंस है वो मैं एक बार लिख के आपको बता देता हूँ। फिर यहाँ पे जो दिए गए हैं ह्यूमन हाउसिंग ट्राइका टेट्राहाइमिना, these all are the different type of organisms which have a different type of repeated sequence and the telomere RNA template sequence. What is this telomere RNA template sequence? It is under the telomerase. This is under the telomerase. Okay, telomerase RNA sequence. So this is all about the sequences which you can write. Now, in case of the mammalian cell, there is another way of stopping the telomere, and that is by the help of TRF2 protein. So it is in the mammalian cell. Let me draw for you. In case of the mammalian cell, so we have what? We have a telomerase, and this is our DNA sequence. This is our DNA sequence. Okay, this is our DNA sequence, and this DNA sequence have an extra 3' end. What it have? It have an extra 3' end. So in case of the in beginning also we need a extra 3' end for the binding of the DNA polymerase, but in beginning we seen if there is no extra 3' end, it can be easily the DNA polymerase cannot bind, and then DNA shortening can be happening. So this is okay, fine. Now this region can be shortened. So what they are doing in this case, they are making a loop. The mammalian cell will make a loop by the help of telomerase. So this is our telomerase enzyme. It have a TRF protein. What it is? TRF2 protein. It have a TRF2 protein, and this is our telomerase. This is our telomerase. So this is our telomerase and TRF2. Together they will help in the formation of this T-loop structure. So it will make a loop. It will make a loop over this. This will make a loop like this, and finally this is called as a T loop. What it is called? This is called a 2 loop, and this is done by the help of TRF2. So TRF2 is like a telomere specific. It is telomere specific. Another thing which we have to study, that is one more thing. Whenever we don't want to make a T loop, there is another protein which is called as a there is another protein which will bind at this side. In this case, if TRF will bind at this side, at the telomere side, suppose this is the side where the TRF2 is binding. Okay, what it is binding? TRF1, sorry. So this TRF1 is binding here, and because of TRF1 binding, the telomerase cannot make a loop. The telomerase cannot make a loop, and it will become crying. It should be crying. No, it shouldn't be crying. Okay, it is crying. Okay, why? It cannot bind. What it is? It is a telomerase. So telomerase regulation can be done by two proteins. What it is? That is a TRF2 and the TRF1. So this TRF2 and TRF1 regulate telomerase binding. Regulate telomerase binding. Hope so you understand this. Now, Drosophila have a different type of DNA, different type of mechanism from preventing from the DNA shortening. Okay. Now that is a dependent on the transposon. So the telomerase protein, whatever the telomerase protein subunits are there in the telomerase, that is some of the telomerase protein are similar in the transposon activity. Okay. So if you see telomere replication in Drosophila. So telomere replication in Drosophila, which is different from the non-telomerase replication. Okay. So our telomerase have some similar to the what I am saying transposon activity. Okay. Transposon. And which transposon? Non-LTR. Non-LTR transposon type of telomerase activity is there in some other telomerase. So, because by the help of this telomerase activity, transposon activity, Drosophila is switched from natural telomerase, normal telomerase to a different type of activity which is dependent on the transposon activity. So, what do you mean by transposon? The jumping gene you must have known. So, in transposon, this is a DNA, and this DNA will make a RNA, and this RNA will be again reverse transcribed, and it will form a new DNA, and this DNA will be switched again at this side. Okay, and it will extending the DNA length. So this is the DNA transposon is happening. This is a basic idea. Some proteins are also involved, which we are not going to talk about. तो यहाँ पे तो ये बात समझ में आ गई कि टेलोमरेज ट्रांसपोजन की तरह काम करता है, और जो टेलोमरेज का कुछ प्रोटीन है ट्रांसपोजन की तरह काम करता है. इसी बिहेवियर को लेकर ड्रोसोफिला ने टेलोमरेज को छोड़ के एक इंडिपेंडेंट ट्रांसपोजन का काम किया, जो कि इस टाइप के जो ट्रांसपोजन है यहाँ पे ड्रोसोफिला के अंदर ट्रांसपोजन सीक्वेंस है. सो ड्रोसोफिला, राइट इट डाउन, ड्रोसोफिला डोंट हैव टेलोमर सीक्वेंस. ड्रोसोफिला के पास क्या नहीं है? टेलोमर सीक्वेंस नहीं है. उसके पास क्या है? इट हैव ट्रांसपोजन सीक्वेंस. ड्रोसोफिला हैव retrotransposon. Okay, what it is? Retrotransposon. And this retrotransposon is what? It is actually third AHR usually telomere will repeat almost 20 times, but this retrotransposon have longer in length. It will repeat around 6 to 10 kb in length. So their repetition is more longer than the telomere. अब इसका repetition समझ के ज़्यादा है, तो यहाँ पे telomere से ये थोड़ा सा ज़्यादा efficient हो सकता है, बट यहाँ पे कोई actual में authentic practical proof नहीं है. ये मेरा अपना समझना है, ठीक है. तो अभी कैसे टेलोमर जो ट्रांसपोजन है ये बताओ मुझे कि कैसे हेल्प करेगा टेलोमर या डीएनए शॉर्टनिंग को. डीएनए शॉर्टनिंग से बचने के लिए ये ट्रांसपोजन कैसे हेल्प करेगा, कॉन्सेप्ट आपको समझ में आ चुका है, you know the concept. You can tell me how this telomere this transposon is going to help the preventing the DNA shortening. So suppose this is a DNA and it have a extended 3 prime and suppose this is a TARD sequence. So this TARD sequence will form what? This will form a RNA and this will be from 3 prime to 5 prime. Now this RNA which is formed, this RNA will go and it will bind here. It will again go, it will bind. Now if it is binding here, then this is complementary, it will easily bind, and then again new nucleotide will be synthesized. Suppose I am giving a sequence T, 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 G. Sorry, it is a RNA, so T will not come, it will come U. U, 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 G, C. Then here it will come A, C, G. So this is a new DNA strand. This is a new DNA strand. Okay. So this is how the DNA replication will be occurring. And now I will tell you the important thing. The HETA doesn't have a reverse transcriptase gene. So HETA, this gene doesn't have a reverse transcriptase gene, but other have the reverse transcriptase gene. So hope you understood this video. If you have any doubt, then you can ask in the comment or you can join our group, which links are given in the description box. The previous video links are also given in the description box and the I button, you can check. Now this is the time where we have to discuss about the disorders. So basically I am going to tell you the disorder related to the DNA. So these are the disorders which we are going to talk now. First disorder which I am explaining, that is a Bloom syndrome. There are a lot of disorders, but I am going to cover few only. So Bloom syndrome is autosomal recessive disorder, and it is because of the loss of function mutation, and mutation is biallelic in nature. So wherever it is a recessive gene, in that this disorder will be seen. Actually, because of the loss of this enzyme, that is a RAG-Q, which is involved in the DNA unwinding, ATP dependent DNA unwinding of a DNA during the DNA repair. Actually, this DNA repair is done by the homologous recombination, and it is also involved in the DNA stabilizing form. So that is a essential enzyme for the DNA repair. If this enzyme is mutated, the following symptoms you can see in this Bloom syndrome. What are these? Red telangiectatic facial rashes occur in the butterfly. So there will be small patches will be formed, which will be little reddish, and it will form like this. Okay, it will form like this. You must have seen in the small children, like in teenage or below 10 years. Also, their head will be small, their height will be small, and it will form a standard deviation from the mean. So if you will see, if this population is increasing, there will be a drastic change in the graph of the population. So another thing is that it will increase the risk of the cancer, as well as it is decreasing the immunity of the human, that is to person. Okay, so this is a important thing. Now we will talk about the different one, that is a Werner syndrome. In Werner syndrome, I will write it here, then I will describe. So this is the another disorder which is related to the DNA replication, that is a Werner syndrome, and it is also the same type of the RAG-Q enzyme disorder. Okay, so in this Werner syndrome, what is there? That is a prokaryotic in nature. What is the meaning? It's a early aging, rapid aging, rapid aging. So you will see this rapid aging in the patient. There will be whitish here in the early age. There will be like loosening of the skin, and you'll find short height, and whatever the features you are seeing in the aged person, all the features maximum be seen in this patient. So occasionally it is because of the missense mutation, and again this is a loss of function mutation. If the mutation is occurring, the function will be lost, And other than the early aging, rapid aging, the symptoms are the like premature heart disease or the strokes, atherosclerosis will be there. And aging and osteoporosis and diabetes are the main symptoms of this Werner syndrome. So Werner syndrome means actually these are the uh, symptoms, but the main symptom is the rapid aging. Now, the important thing which we have to understand how we can use this for our knowledge purpose. Why we studied these all things and what is the conclusion. So in this thing, you have to give a hypothesis. Suppose I will give you one example, live example. So this is a research paper from a research paper. We have a two type of cells. I suppose if we have a normal cell and the patient which have a cancerous cell. So in the cancerous cell, there is a rapid replication of the DNA is continuous. And this is in normal cell, there is a programmed cell replication.